Well, good evening, everyone, and welcome to Science on Tap, our first one of 2021. We're really excited to be back and for tonight's speaker. And I am especially excited because I got my first dose of the COVID vaccine today. So big thank you to Montana for providing it and to Tribal Health for making sure our community here in Folsom is vaccinated. Just real quickly, the Flathead Lakers are a nonprofit that's devoted to conservation and advocacy of the Flathead Lake watershed. And I'm recognizing a lot of familiar faces, but if you're not a member, be sure to check out our website and join. We work closely with the Flathead Lake Biological Station in educating folks and advocating for good water policy for this watershed. And we're also excited to hopefully, fingers crossed, open our doors this summer and invite you all into our office. This is our 5th year for Science on Tap, and we liked having this monthly event to bring together folks from all walks of life and scientists in the community and give them an opportunity to share their research and their knowledge with us in a candid format. And my dog is really excited about Science on Tap in the background. So I will hand the mic over to Sean.
All right, thank you very much, Kate. I'm gonna go ahead and minimize that there. Perfect. Well, yeah, it's really, I'm glad to be back on Science on Tap. Jay, you're spot on. You said earlier, it's a shame we're not all together. Fingers crossed soon, like Kate was saying, and we'll get there with vaccines, I'm sure. Thanks to the good work of certain scientists that we might have in the room, right? So I wanted to take a couple minutes and go over Science on Tap. We've been on a touch of a hiatus, but that's, given the times and given how much Zoom I personally am on, I find it an okay respite. I think this is hour 7 on Zoom for me today, so it's a lot, but we're doing all right. I am really excited about tonight's speaker, but I'm even more excited, no offense, Jay, but I wanted to talk to you a little bit about the Science on Tap website. So I'm going to go ahead and share my screen for a moment and just let you guys know that the Science on Tap Flathead.org has been completely revamped and it looks incredible. I'm just giving you the slow, I don't know if that makes people sick at home or what, but I'm just kind of giving it there. What I want to next focus on is this, that I'm actually really proud. I don't want to, I'm going to get choked up. The Science on Tap Flathead YouTube channel, it says this channel doesn't have any content, but it's lying to you. We need to fix the home site there. We have a lot of videos thanks to the hard work of the Flathead Lakers intern, the media intern from Eric, thank you. Eric did a great job on a lot of these, and looking through the book sheet here, the police blotter, I guess. We've had an amazing assortment of speakers from fisheries biology through sewage treatment management, avalanches, beer, wolves, loons, it's nuts. I'm just really proud of the fact that we have had such a breadth of content. We've had climate change specialists, Nobel laureates, and members of the Academy, and it's just incredible that tonight we also get to have a major driver of what has become a major, I would say a proud and a crown in the jewel of University of Montana research, which we're going to learn more about today. So I'm going to argue that everybody should probably spend at least probably the next 12 to 15 hours binge watching all the YouTube, all the previous episodes, but not until, I know it's going to be tempting. Don't do it. Not until we're done with tonight's event, which I am really excited about. So Dr. Jay Evans is going to talk to us about the research he's done. But if you didn't read the headline, we're really kind of, I'm really proud to be a colleague and an associate even of this kind of work. And I'm excited that we get to talk about it tonight. And I hope that we can have a good amount of questions regarding sort of the questions of how to talk your buddy who doesn't want the vaccine into getting the vaccine. So I'm going to prod for those kinds of questions. You know, I love those kind of how to get out of climate, you know, change conversations in three sentences and you just win the argument. Some points like that would be great. So without further ado, I'm going to stop sharing my screen and introduce Dr. Jay Evans, University of Montana.
Thank you, Sean. Let me start sharing my screen then. You see that all right?
Looks good to me.
That's good. Okay, great. No, I really appreciate the invitation to come and speak tonight. I've talked a lot, as you might imagine, in the last few months about COVID. The presentation tonight kind of has a mix of stuff. It talks a little bit about our Center for Translational Medicine at the University of Montana. Full disclosure, I'm also president and CEO of a biotech company in Missoula called Inimune Corporation, where some of the assets we're developing at University of Montana are now moving into phase 1 human clinical trials. As you'll see, I actually have a strong connection to what this group does in fisheries, and I'll talk just a little bit about that. But mostly, I'm going to talk about the 3 approved COVID vaccines, a little bit about the safety and efficacy data, and then I'll finish up with a little bit of information about the UM's COVID vaccine research program and about our team. I only have about 40 slides. Hopefully, we'll get through this in 30-40 minutes and have lots of time for questions about anything. A little about me. You see, there's actually a lot of water and fishing on this slide. I actually have a pretty long history in conservation. I've spent about 10 years on the Bitterroot Trout Unlimited board of directors. Did series just like this down in Hamilton, Montana, seminars on fisheries and clean water, aquatic invasive species. I'm currently on the board of the Montana Bass Nation, which is a bass fishing organization. As you know, there's a lot of bass now in Flathead Lake. It's becoming a very popular spot for some of the local anglers in the state. And you know, avid in the outdoors with me and my family, I always have been. Very familiar with stuff going on in the state as far as aquatic invasive species, what's going on in Noxon, what's going on with mussels. Just about 2 or 3 weeks ago, I was down at Lake Havasu. My boat is now drying in the garage because they have mussels down there. So happy to talk about my thoughts on that and invasive species if people wanted to ask questions on it. And the University of Montana's picked up on this and did a Grizzlies in the Wild segment about how COVID research is similar to fly fishing, which is kind of a strange combination. So a little bit about our group at the University of Montana. We're a big, fairly large vaccine research and discovery development team. Our group was founded in 2016 when 15 people came up from GlaxoSmithKline in Hamilton and founded this new group. Since that time, it has grown exponentially. We now have over 60 researchers in our group working on vaccine research here in Missoula. That's between the University of Montana and our group over at Inimune. And we've brought over $97 million of new federal grants and contracts to the university since 2016. So it's been wildly successful beyond our wildest imagination for where we would go. We do work on a lot of different vaccine programs. Of course, we'll talk a little bit about SARS-CoV-2 or COVID-19 today, but we also have programs for universal influenza virus, tuberculosis vaccine, Pseudomonas E. coli, MRSA, whooping cough vaccine for pertussis, Lyme disease, immuno-oncology, allergy. And we actually have a program now that we're going to do 2 phase 1 clinical trials and a vaccine targeting opioid addiction or fentanyl abuse. And actually, next week, there's a community lecture series I'm going to give at U of M talking a little bit more about that program. It's kind of a unique program. And as I said, when we started this about 5 years, closing on 5 years ago now, we started with a strong public-private partnership between the university and a local company here that we founded in Missoula called Inimune. And this public-private partnership in a place like Missoula is why we've been able to grow this program to where we are now. And it's been a lot of fun along the way. And we've met a lot of great people in the community that have helped us get this done. And we also can't do this alone. You can see the connections just within the US. We have a lot of international connections too. All of the people on the screen here are involved in our various vaccine research programs at the university. We have collaborators all over the world, across the US, and a lot of our technologies are developed in partnership with a lot of these great other institutions that we've developed partnerships with. A little bit about vaccines, just kind of jump up on my soapbox and talk about the benefits of vaccines. They really are a fundamental driver in global health. There's only one medical invention, called a medical invention of all time, that saved more lives than vaccines, and that's clean water. Each year, vaccines prevent 2 to 3 million deaths, and many children are saved from disabilities. In 2018, it's estimated about 86% of the children under year 1 receive a DTP vaccine, which is a pretty amazing accomplishment. Despite all this, there's still millions of children that die annually from vaccine preventable diseases. And of course, there's new emerging diseases, which we're hearing a lot about now. So of course, we can do a lot better. A couple of the highlights of what vaccines have achieved so far. Things like, smallpox has been eradicated, polio, worldwide incidence has decreased, more than 99%, measles, DTaP, and the list goes on. When you look at the list of vaccine preventable diseases, you can see now COVID-19 is at the top of this list, just added. It's no wonder people get tired of vaccines, especially children. Seems like they're getting immunized every 3 months or every 6 months against something. The list is long. But with that said, the success that's been seen in vaccines is hard to argue against. You can see the incidence rates of things like measles and polio have dramatically declined since these vaccines were introduced. So I'm going to jump now and talk a little bit about SARS-CoV-2 vaccine. This is a photo here of Sandra Lindsay. I remember this day. She was the first person vaccinated after the trials finished with the approved vaccine from Pfizer. It was kind of a monumental day and started hopefully what will be a new era of being able to reopen and not have to do everything via Zoom. And so, what does the daily confirmed cases of COVID look like in the U.S.? I've been giving these talks now for 6 months, and I keep updating this graph. Boy, when I was giving talks in November and December, it was pretty bleak. Looking at what was going to happen to the mortality rate as we move through the next couple of months. Fortunately, whether it's the seasonality of coronaviruses, which they are seasonal just like flu, or whether it's people stepping up and doing social distancing or masking, we don't know. But fortunately, the case rates, daily new confirmed case rates in the United States is really dropped in the last month. How has Montana done? Very similar. We're following the national trend very well. Peak case rates happened in the middle of November. People can say, oh, the holiday season is what caused it. You can see it November 17th. It happened before the holiday season. If anything, during the holiday season, we were careful, and we have seen a drop off in the daily new case rates, fortunately. And it seems like Montana, if you look at it today, I think the newspaper said 50, but of course that might have been from reduced testing over the weekend. But it appears as though the curve is in the right direction, which I think is fortunate for everybody. And so, the other thing I like to talk about with vaccines and treatments is the response to COVID has really been unprecedented. I gave talks last year in February about this, and I went on record as saying, how long is it going to take to get a vaccine? I said a minimum of 5 years. I would never believe that last week I got my mRNA COVID vaccine. I would have never believed a year ago that was possible, given that mRNA technology was only in phase 2 at the time. You can see here the number of new antivirals, treatments, and vaccines that have started in the preclinical discovery through clinical phase. And it's just astronomical. I've never seen anything like this in the over 20 years that I've been in the vaccine industry. And you can see there where the UM's COVID vaccine program started. We started working on this about this time last year. February, March of last year is when UM started their COVID-19 vaccine program. And to put this in a little bit different perspective is, how long has vaccine development taken? You see here SARS in 2003. How long did it take to get something to the first human study? It took 20 months. As you can see, that timeline has gone down, down, down. And look how quickly we were able to get a COVID-19 vaccine into phase 1 clinical trials. And people, one of the biggest fears people say is, we did it so fast, how can it be safe? How can it possibly be safe? There's a lot of concern over the safety of the current vaccine, and I'll show some of this. But, you know, vaccine safety has always been the #1 priority of vaccine companies and developers from day one. When we're developing new, brand new vaccines at the University of Montana, whether it be an opioid vaccine, a COVID vaccine, TB, or flu, our #1 thing is safety. We identify vaccines that maybe don't have the best safety profile very early in animal studies. They never make it anywhere close to a phase 1 study. And then phase 1, 2, and 3 is all focused on efficacy and safety, and making sure that, unlike a lot of treatments for whether it's on oncology or diabetes or other things, vaccines are given to healthy people. Most treatments are given to someone that already has a disease. You can really weigh the risk benefit. But for vaccines, you're giving it to people that are healthy that you don't even know that they're ever going to get the virus. So they have to have a very clean, squeaky clean safety profile. And the FDA insists on this. And this hasn't changed with COVID. The same safety precautions are in place with the COVID vaccines, or have been in place with any vaccine that's been approved in the past. And if anything, they're even higher now. And the reason we were able to shorten it and get a vaccine out in a year is because the regulatory red tape was cut. Not the safety red tape, but the regulatory. So companies were able to submit their phase 2 and phase 3 clinical trial plans for the FDA review before their phase 1 clinical trial was finished. To really shorten the window of time between phase 1, phase 2, and phase 3, so that a week after the phase 1 ended, a phase 2 could start. And the only way the FDA was able to do this was by dropping everything else they were working on. A lot of other clinical trials have stopped, completely stopped, because the FDA wanted to focus on COVID and make sure a vaccine was delivered that was safe. So there's 3 main types of COVID vaccines that have been in development and are now approved. #1 is the type of vaccine we're working on at the university, and this is what they call a traditional protein-based vaccine. So the spike protein, which is on the surface of the coronavirus, is made recombinantly in cells in the lab in a tissue culture dish. It's then purified, and that's what's used as the vaccine. And at the University of Montana, we had something called an immunostimulant or an adjuvant to drive a better immune response. Examples of these types of vaccines, we just heard that GSK and Sanofi just entered phase 2 clinical trials with their vaccine with an adjuvant called ASO3, an immunostimulant in the recombinant protein. There's a company called Novavax, which has a matrix M adjuvant to boost the immune response that's currently in phase 3 clinical trials. The second type is the type that was just approved this weekend, and that's a type that's the J&J vaccine, the 1-dose vaccine, based on an adenoviral vector. So they take the same spike protein, but rather than using the protein, they use a piece of DNA that encodes for that protein. They put it into a vector, which adenoviruses infect people, common cold virus, things like that, but they make it so it's replication incompetent. It won't infect you, it just gets into the cell. And then produces that spike protein, and then your body can respond to it as a vaccine. The third type are the currently approved ones from Pfizer and Moderna, and that's an mRNA vaccine. So you have an mRNA that still encodes for the spike protein, gets packaged, and then that expresses the protein, and you generate an immune response. So all 3 of these technologies are based on generating a neutralizing response against the spike protein from the COVID virus. There's just different ways to get there. So a little bit more detail about how the Moderna and Pfizer vaccines work. This is the vaccine. Anyone in the room who's been vaccinated, this is the one you receive. And I would say it's the one you're most likely to receive, but now that the J&J vaccine is out, single-dose vaccine, we're going to see that being widely available too. Again, they make this mRNA sequence synthetically based on the sequence from the spike protein, which is on the surface of the coronavirus. mRNAs are very unstable, so they coat it in a lipid coating, a lipid coat around it, and that's the vaccine that gets injected. Because of the nature of the RNA and the lipid coating, these aren't very stable vaccines. That's why they have to be maintained at -8 at really extreme temperatures to keep them stable. But they are very effective. This technology had been advanced to phase 2 for an influenza vaccine and an RSV vaccine candidate. So it's not like the regulatory agency didn't have any experience with it. It had just never been in phase 3. Because it's a fully synthetic vaccine, that is, everything is made synthetically. You don't have to have a cell line make it for you. That's why this was the first technology to get to the clinic, because all they needed was the sequence. As soon as they had this, as soon as Moderna had that sequence of the spike protein from the isolate from Wuhan, China, they were able to start manufacturing their vaccine long before COVID was ever in the US. And that's why that vaccine technology moved so quickly. A little bit about the Pfizer vaccine trial. This data's been out now for a little bit. I want to, kind of dispel some of the questions that came out, especially when this vaccine was first released. The first thing I heard was, well, yeah, but they didn't test it in people that are most susceptible to the hard effects of COVID. Those are the people over the age of 65. When in truth, the Pfizer phase 3 study, 26% of the people in that vaccine phase 3 study were over the age of 65. There were also minorities included. So they did really take a good swath of the population, men, women, and people over the age of 65, those most susceptible at risk for COVID. Next is efficacy. We've heard a lot lately is, why not just give a single dose of the Pfizer vaccine to everybody so more people can be vaccinated? Clinical data from the US clinical trials show that after a single dose, you were only about 50% protected. After 2 doses, you get the 95% protection, which argues against using a single dose and getting as many people vaccinated as possible, because you still might be 50% susceptible to COVID. So a little bit on safety data, and I'll get into this a little bit more. A lot of people are concerned about safety with these new vaccines. This looks at the data from the phase 3 study, looking at, within 7 days after the vaccine, local injection site reactions, local pain, redness, swelling, versus systemic effects, fever, fatigue, chills, joint pain, headache. These are pretty high rates. 75% of the people that get the COVID vaccine should expect to have a local site reaction. My arm was sore over the weekend. I knew I'd gotten the vaccine. There was no question about it. And fatigue, I felt tired of it. I got my vaccine on Friday and I was tired Saturday and Sunday, which tells me my immune system was working. I didn't have any fever, chills, joint pain, but my arm was sore and I had some fatigue associated with it, clearly. But the main thing is there's no indication of antibody-dependent enhancement of disease in the clinical trial, which was a concern with this type of vaccine. And the severe adverse events related to COVID were no different from any other vaccine on the market, and they were actually less than some of the other vaccines out there. So the severe adverse events were generally in line with the placebo controls for these phase 3 studies that were on the order of 40,000 people. So they were also very large studies. So I put this table together to kind of compare what a seasonal influenza vaccine adverse event looks like in relation to the Pfizer COVID vaccine on dose 1 or dose 2. So here are the two mostly used seasonal quadrivalent flu vaccines. One is Fluarix and the other Fluzone. You can see injection site pain. About 30 to 40% of people report having injection site pain, within days after the vaccine. For COVID, that's 80%. So you can see it's about twice as many people report pain at the injection site. If you look down to the systemic effects, about 20% of people say they have a headache after getting a flu vaccine. 15% of people say they have muscle aches. If you look at the COVID vaccine, it's about twice. So I think that's, so I would say it's about twice as reactogenic in that setting as a seasonal flu vaccine. But the severe systemic adverse events that happen are equal to or less than the flu vaccine. So you can expect to have these types of responses if you get the mRNA-based vaccine. But generally they're transient and they go away in a day or two. So there was just a vaccine approved this weekend, the Johnson & Johnson or Janssen vaccine, which was exciting. The VRBPAC meeting for FDA, the advisory board meeting, was on Friday. I have a friend that was in that meeting. It went really well. And then the vaccine was approved over the weekend. As I said, this is a different type of vaccine. It's based on an adenovirus 26 vector. It'll be the first vaccine approved of this kind as well. Again, this vector gets into the cell. It's a non-replicating, non-integrating vector. It just goes in, expresses the spike protein, which your immune system can then respond to, giving both a cell-mediated immunity or cell-based immune response, as well as an antibody-mediated response. What does the efficacy look like for this vaccine? You probably heard about it on the news over the weekend. It's active actually within 14 days of a single dose, which is pretty exciting. It's a single dose vaccine. 28 days after the second dose, it's 72% effective. What's really exciting is it can be stored at 4 degrees for 3 months or -20 for longer periods of time. This is going to be a game changer for countries that don't have -80 freezers, which is most of the countries other than US and Europe in the developed nations. For non-developed nations, having this type of a vaccine changes everything, because now you can use normal refrigerated cold chain, get it to them within 90 days, administer the vaccine. It's gonna allow us to really vaccinate more of the world population than was possible with the mRNA-based vaccine technology. So people ask, so which one should I get if I'm offered the vaccine? Here's some comparisons. Here's the, if you want to look at reactogenicity or local injection site reaction, systemic responses, you can see compared to the flu vaccine, again, about 30–40% of people report local injection site pain. It's quite a bit higher with the mRNA vaccine than it is with the J&J vaccine. J&J vaccine looks more like the traditional flu vaccine as far as the local and the systemic adverse events. You see maybe a little bit higher for muscle aches and headaches versus the influenza vaccine. So I would say, a few less effects with J&J vaccine versus the Pfizer or the Moderna mRNA vaccine, but really still comparable on the bigger scheme of things in relation to the flu vaccine. One thing of note is that, which they didn't report in the Pfizer trial because they had 95% efficacy, even though they only show 66 to 72% efficacy, there's 85% protection against severe disease. So I would say if people get the opportunity to get either vaccine, you should get it. Herd immunity is a real thing. These virus mutations and variants that are out there are a real thing. They're a concern. The more quickly we can get the greater percent of the population vaccinated, the less likely we're going to have these variants pop up that might get around the vaccine. Of course, we're also facing issues with vaccine distribution and with hesitancy to take the vaccine. During the last election cycle, people were arguing that about half of people said they weren't going to take the vaccine. I haven't seen results lately, but from the lines and people wanting to get the vaccine, my guess is it's a lot higher than 50% now that want the vaccine, now that it's been widely distributed and they see how safe it is. Where are we at today on vaccines? About 37 million of the Pfizer vaccine and about 35 million of the Moderna vaccine have been administered to people. Where are we as a state? You can see from per 100,000 people, we're doing pretty good. We're one of the darker blue states, which means per 100,000 people, we vaccinated more than a lot of other states. So I think Montana's doing pretty good at getting the vaccine rolled out to the people who need it, which is great to see. Another big concern is are we continuing, once this vaccine rolls out, how do we continue to monitor safety? Because you really don't know how effective and how safe a vaccine is in lots of different subpopulations of people unless you have great after approval safety monitoring. So this slide summarizes the post-approval vaccine safety monitoring that's in place for COVID. I'm not going to go through all the different surveillance systems, just to say it's extensive. And we're already getting pretty good detailed reports on safety now that, tens of millions of doses, closing in 100 million doses now have been administered. They're getting really good safety profiles on what this looks like. And people who are pregnant, they're collecting that data now to give confidence to those groups of people to get the vaccine. So how does this look? This is when more than 55 million Americans, I think it's now up to 100 million, have gotten the first dose of the COVID-19 vaccine, how does the safety look compared to the phase 3 clinical study? What they're finding is it's very similar. Remember, I showed you early, 70 to 80% of people report injection site pain. They're seeing the same thing now that the vaccine has been rolled out. This vaccine safety data link reports no increased risk of the 20 most common medical conditions. Because you can't always track a vaccine until you get it into millions of people to understand how that looks in relation to some of the more rare diseases that people might have, and they're tracking that very closely. We heard a lot about anaphylaxis and these studies. Yes, in the phase 3 studies, they excluded people that had a history of severe anaphylactic response to past vaccines. And so they didn't really see them until it was rolled out. Now they're saying it's about 2 to 5 people per million doses has an anaphylaxis response. Compare that to, the risk is, of course, much lower than the risk of potentially dying from COVID. If you have a 3% mortality rate with COVID, for every million people, that's 30,000. Here, they're talking about 2 to 5 people have an allergic reaction to the COVID vaccine versus of that same group, 30,000 that would have died without getting the vaccine. The numbers are clear. And those people that are having allergic reactions are treated there at the site, and they haven't had a single COVID vaccine-related death due to an allergic response. So the data is pretty clear on that. A little bit about our vaccine program at the University of Montana and what we're working on. Like I mentioned earlier, we're working on what we call a protein-based vaccine approach. We take a protein, the spike protein shown in red here from the virus, and we'll inject that in with an immunostimulant to drive the right kind of immune response. That's the system we're doing. So we're working with a group over at Mount Sinai Medical Center in New York, who has the animal challenge model up and running for COVID, because they have a biosafety level 3 lab where they can do that there. They were also one of the first groups in the country to start producing the spike protein and getting the challenge model up and running. And so University of Montana has been contracted by NIH as a vaccine testing center for the COVID vaccine. So right now we're looking at about 8 different vaccine technologies developed at different universities and countries. And really what we're trying to do is find the next generation COVID vaccine. We all hope it won't be necessary. We all hope the current vaccine solves it and we can eradicate this or diminish it to a level or we can go back to normal. But just in case the durability of the response isn't long enough, the duration of the response isn't broad enough, if these variants that are circulating can get around the current type of virus technology. Our group works on influenza and TB and pertussis because we improve current vaccines by adding different types of immunostimulants to it. So we're already doing that with the spike protein in COVID to have a next generation vaccine ready to go if it's going to be needed. So how do we do that? We take the spike protein from the virus, we produce it recombinantly, purify that, and then add immunostimulants to it that drive the right type of immune response that we want to get against COVID. We've been doing this for years. This is kind of how the vaccine technology and industry has moved away from whole inactivated, whole vaccines that are very reactogenic to very subunit defined vaccines and immunostimulants that are safer and more effective. And we do this by adding what's called adjuvants. So what are adjuvants? They're a component that we add to a vaccine to generate a stronger immune response. They help in the breadth of that response. In other words, we have vaccines that will likely protect against these variants because it broadens the type of immune response you get against the pathogen, as long as the durability or duration of that response will last. Let's say we find in 3 years that everyone has to get vaccinated again because your immunity wanes or drops off over time, like what happens with pertussis and the seasonal flu vaccine. We have adjuvants that will boost that response, as you can see in the green line here, and then you get a much longer lasting durable immune response. There's already adjuvants in use in vaccines. Aluminum salts is the most common one. ASO3 here, oil and water emulsions, is the adjuvant that's in GSK and Sanofi's COVID vaccine. It's also approved in a pandemic influenza vaccine. And there's a number of other types of adjuvants. And so, you know, a little bit about our vaccine. Again, we're targeting the spike protein or the S protein from coronavirus, from COVID. We're also looking at the what's in green on the slide, which is what they call the RBD, or the receptor binding domain. That's the part of the spike protein that actually binds to the receptor on the human cell, allowing the virus to get in. So that's really where we're focused on getting an antibody response against, so we can neutralize that virus getting into the cell. We also do a lot with formulation and delivery. I'll skip over this with time. There's many different ways you can deliver these protein vaccines to get the immune system to recognize them and stimulate the right kind of response. And so this is some data. I've just got a couple of data slides, and then we can jump right to questions. This is a study done in mice right here at the University of Montana, where we take this receptor binding domain from COVID. We inject it into mice. You see there the black circles. That's the antibody response that those mice would get. 3 different adjuvants, 1, 2, and 3, that were designed and developed and invented at the University of Montana were added. And you can see this is on a log scale, how much we're able to boost the antibody response in these mice to that receptor binding domain to COVID. So you can imagine in a person getting that high of an antibody response by adding an immunostimulant is really beneficial to neutralizing the virus and providing a long-lasting protection. Here's another example. In this case, we're using the entire, what they call the full-length spike antigen, the full-length spike protein. This is similar to what's being done with the approved vaccines, use full-length spike. You know, in blue is just this full-length spike protein alone. Again, antibody responses for mice. And then you can see whether it's adjuvant 1 or adjuvant 2, different doses of the antigen, doesn't matter. All of these are able to generate a really strong, what they call humoral or antibody-mediated immune response against COVID. So these vaccines now are over at our partner institution over at Mount Sinai, New York, being vaccinated. Mice are being vaccinated, and they're being challenged with COVID to see if we're able to neutralize and protect mice from that response. And finally, one benefit of having this type of a vaccine adjuvant is you can also generate what they call a cell-mediated immune response. And this is the kind of immune response that gives you long-term protection. Antibodies over time will go down in your immune system, but the cell-mediated response can be around for a lifetime. As you can see here, again, the cell-mediated response in blue to the antigen alone without an adjuvant is basically non-existent. It doesn't happen. Whereas adjuvant 1 in the green circles, you can see drive a really strong T cell response against this protein and drive probably a long-lasting memory response in these mice, long-term protection. Adjuvant 2 here in the purple, while it did give a good antibody response, didn't give much of a cell-mediated response. And then the fifth one is actually, the last one is actually adjuvants 1 and 2 together. So you can see we empirically work through these different systems, delivery systems and adjuvants at the University of Montana to identify the best COVID vaccine. All of this data is then being supplied to the National Institutes of Health. And so when a second generation SARS-CoV-2 vaccine is needs to move into phase 1, we'll be able to advise them on the best protein adjuvant combinations for this type of a vaccine approach. Like I said, hopefully it's not needed, but if it is, we'll be ready. And you know, we'll see where the current vaccine strategy goes, but I wouldn't be surprised to see this move into phase 1. So the NIH has it ready and has good safety data ready in case it is needed. At the same time, we're generating a lot of useful data for the next pandemic, whether it's flu or whether it's Ebola or something else. We're learning a lot about how to develop vaccines quickly and safely and effectively for worldwide distribution. And so that's it. That's all I had for today. I know I went through it pretty quick. I wanted to make sure there were lots of time for questions. Whether those questions would be about our COVID program, about current COVID vaccines, about contact tracing and COVID testing, or about aquatic invasive species. I'm happy to answer questions on that too.
Well, thank you very much, Jay. That was really great. I think I was blown away by the fact that that you were so proactive in terms of, as soon as there was a problem in China, like there must have been an awakening, or not an awakening, but just it struck me as your response was so, not just your response, but the response was so fast that it's almost as if folks are ready for the next outbreak of respiratory virus in particular, and that there's I guess my question is that the case? Is your research and other researchers kind of hanging on and waiting for these outbreaks and the next type of pandemic, potential pandemic virus to come along, and very proactively responding to false alarms? And that that drives a lot of the sort of competitiveness in the market and whatnot, or what enables the ability to respond almost so far ahead of the U.S. curve, so to speak? And yeah. I think you get the gist of my question.
I do, and it's a great question. It's obviously complex. People ask me, gosh, if we had, because there was a MERS outbreak before, there was a SARS-CoV-1 that happened before. Why weren't we ready for this? Why weren't we more ready for this? Why didn't we already have the mRNA vaccine technology for COVID through phase 3? Why hadn't it been tested? It's a great question. If I actually didn't have it in this presentation, I've got a map of the worldwide emerging diseases. And if you look at the map, it's hard to see the world because there's so much writing on it. I mean, there are 100 different diseases similar to COVID that we're watching. And which one is going to come out when and what timing is really hard to predict. As you can imagine, when MERS and SARS-CoV-1 came out, there was a lot of interest in it. There was a lot of research put into it. But when it went away, they went back to other things of greater importance at the time. Things like flu, TB, malaria, opioid addiction, oncology, HIV. There's a lot of current diseases, pertussis, things that are affecting people today. And that's what the community wants to see the dollars put towards. And rightly so. But we need to be poised to react when something like this happens. So the reaction was unprecedented. We started working on it, in February of last year, which is crazy because that's before it was really even in the US. But we were watching what was happening in China, and you could see that it wasn't going to be contained. Before it was here, we knew it wasn't going to be contained just because of how it was spreading and what was going on. And fortunately, as you can see, we have a lot of funding from NIH. But we do some pretty big NIH contracts. And so we got on the phone with our contract officer and said, hey, what do you want us to do? And he says, we need you to divert money and effort to this now. And then we will put supplements in place to get you the money back. And so we were able to move efforts immediately from some of our programs like on TB and influenza over to COVID pretty much overnight, start working with this group at Mount Sinai, which were the first to produce the recombinant antigen, and start getting the help pay for them to get the animal model testing up and running. And then in the meantime, we were able to work with NIH and put about $5 million in supplement monies coming into the University of Montana to pay for that work we'd already started. So it happens really fast because we're a contractor to NIH, not just a grant awardee. So
that makes sense, given the potential spread and whatnot. I guess it strikes me even that the symptoms that can be remarkably vague to across the board get identified as these emerging diseases so quickly, even when within a single, like you said, that it surprises me there's so many maps or so many blips on the map when like fever is probably common, among them all. It's like, wow. Yeah. And I guess it's amazing to think about how quick the how quick things were able to, or how quick the government was able to respond in terms of funding, and how quick the researchers were able to put their expertise to use. So I got the first question in. I think the best way to do it with the group tonight, where if you have questions, I'd encourage you to write them in the chat to me, directly to me instead of to everybody, just straight to me, and I'll sort of divvy them up, or translate, I suppose, and if I get it wrong, let me know. And that will make I think that'll make things go a little smoother, or with less interruption or herky-jerkiness. Yeah, I know. I guess I'll ask another question while folks kind of start to send them in. And but what strikes me is, how are you able to continue your work when for all intents and purposes the university was shut down? I mean, were there special circumstances, considering you're trying to fix this for everybody, and did you find the balance between those, being safe and we were really lucky. The response at the biostation was amazing. We had a protocol in place. It was number one priority was safety, and everything else kind of had to be put into place under that umbrella. And if it didn't hold that umbrella up, it didn't hold up, right? And the protocols were designed to continue productivity, but there's no doubt it was hindered. So how did you balance that in your lab?
So that's a great question. So we were almost immediately given frontline essential worker status, research worker status at the university. So we never saw any delay at all. We've stayed working at the labs of the university through the entire thing. We were early adopters of mask wearing, mandatory mask wearing, and our group was earlier than the city and the county and stuff put on those types of regulations. Daily cleaning protocols. We started allowing people to do any book work. And, as a matter of research is a lot of writing in notebooks, a lot of experimental design, a lot of meetings. All of our meetings went to Zoom really quick. And all of if you didn't have to be in the lab, you worked from home. But people that you can't do lab work at home. You can't. And so we kept working. We set up a pretty early surveillance system. One of a Missoula testing lab here called Fire Diagnostics. We set up a contract with them, and I had COVID test kits in my drawer. And if somebody came in, sniffly nose, achy, we said, don't come to work, period. We'll have someone else do whatever you had to do that day, and we would arrange for them to come in and get a COVID test done. We did our own contact tracing and tested another group of people. And I'm happy to say that doing that, and I think the mask wearing, washing hands, being cognizant of social distancing from early on had a big impact. I think we've had 5 or 6 positive COVID cases within our team of 60, which is about where we expect to be. We have not had a single case of workplace transfer within the team because of those. It just shows that if you socially distance, we're reasonable. We had a person who tested positive for COVID. It happened to happen in the afternoon. The person started feeling sick in the afternoon of one of our big harvest days for one of our mouse COVID experiments. This person was shoulder to shoulder with other people for hours, but they were wearing masks. They were taking precautions. They, washing their hands, being careful, and they didn't transmit it to anybody. So it just shows that those general basic precautions that the CDC and the county and the state have been, telling everybody for a year now, do work if we do them right. And so that's what we've done, and that's how we've kept working. And so far it's been successful.
It's impressive. I think it says a lot if you can follow the rules a little bit. It's helpful. I think in terms of it's good to know that UM and the response that you were able to get in fast tracked, I guess, or put into a place where it was, you've had it figured out, and it was it's a good thing. All right, we got a few questions that are rolled in. First one. So the question is there any data related to resistance and what I assume to be your resistance after having suffered or having contracted COVID? So is that as good as a vaccine? or what are the drawbacks, and what are, sort of expand upon that a little bit?
Good, another great question, and something that unfortunately there's not a lot of great answers to. There are studies that are coming out now. You see published reports all the time about people who had COVID, who have now had COVID a second time. Usually the second case is much less severe, suggesting that natural immunity can wane over time. If you look at across the spectrum of those reports, some of the really early reports that came out of China and South Korea and some of these other places, they actually found were false positives or false negatives the first time around. It actually weren't true reinfections. But now they have cases that have been published on where someone tested positive, they cleared the virus, they were clearly negative, and then 9 months later contracted COVID again. In most of those cases, they were asymptomatic the first time. So they had a very mild case the first time, probably didn't have a really high antibody titer, and it wasn't sufficient to protect them, and they were able to get it again. And because of cases like that, hopefully they start establishing a level of antibody that's required to maintain protection. This is what's going to come out of all of these phase 4 vaccine studies, all the stuff that's going on now. They'll start to understand what level your antibody has to be to maintain protection against the virus. And if someone falls below that, they'll need to get a booster vaccine. But how long that lasts from a natural infection versus a vaccine? Remember, a year ago, the virus wasn't even in the US. So it's hard to say a year from now what that level of protection is going to look like in those people. And so there's not a great answer out there now.
Yeah, that
there's
makes
a lot of people focused on finding an answer to that question.
Right. Yeah, I mean, the fact pointing out that it wasn't here a year ago is something that it's so hard to even recall what that was like. That I think it's so it becomes so you become jaded to the fact that this is a marvel, and that
The other related question that I've gotten quite a bit is people asking, if I had COVID, should I get the vaccine?
Right.
And I'm telling people that it can't hurt. If anything, it gives you a longer, more durable immune response to get the vaccine. If you're 25 years old and healthy, you got COVID, you were actually pretty sick, chances are you're going to be protected for several years. If you want to pass on your opportunity to get the vaccine, to give it to somebody else that maybe didn't get COVID, to get herd immunity, I think that's a reasonable approach. If you're 70 years old, or you have some autoimmune or some other health condition that puts you at risk, you had COVID, but it was a pretty mild case, I'd get the vaccine, no question.
Right. Thanks for bringing that up. That was something that's a good secondary, second part to that question. What are the travel restrictions like after the second Moderna shot? So I don't know how that relates to policy necessarily, or to the vaccine itself, but how long do folks have to wait after their second Moderna vaccine?
So it takes about 2 weeks to achieve full protection after the first dose, and then 50%, and then after the second dose. So I would say if someone was going to travel and they wanted to feel safer when they travel, I would wait for about 2 weeks after the second dose. Of course, you're already 50% protected after the first dose, but I would wait till about 2 weeks after the second dose. As far as a policy and state requirements and masking and all of that, I have not heard of any states allowing people that have a card that says I'm vaccinated to get out of any of the travel restrictions that might be in specific states, or eating at a restaurant, or traveling on an airplane. You're still going to have to follow all those requirements. I think the legal part of that, regulatory part of that, is still being worked out, and how they open that up for people that have been vaccinated, I'm not sure anybody really knows yet.
Wonderful. That was, I think that's one of those things that it's like, man, you hope it doesn't start. Well, I've been vaccinated. I don't know. I think about that scary part of it. We have a few more questions. I'm going to try to summarize one of them here. So it went to the everyone pool so you could see it, but there's some questions about the adjuvants, and if they're the cause of some of the correlative responses in the sort of adverse effects. Or is it? I think is there the things that boost the response, are they are things like fever and fatigue? That's like, you know, is that a sign that it's working? Like a fever is fighting something off. You should, you know, the idea is that the response it's a positive response, even though it's a negative event, right? But how do those things relate? Is it the antigen that's the problem? Is it the adjuvants, or is it just the needle? You know, like what's the deal?
So there's no, I mean, if you take these little small needles that are associated with vaccines, they don't cause much damage at all. It's not the needle. Of course, if you take a half a mil or a mil of solution and inject it into your muscle, there is some muscle tissue damage. That is going to cause, so your muscle would be sore if you took and just injected saline into it, but not to the extent you get with the vaccine. So the antigen itself, the protein component of the vaccine, does add to the reactogenicity or the side effects, but the adjuvants definitely impact that. So you need to drive an immune response to get an effective response against the vaccine, which is why when you get a virus or a bacteria, you feel like crap, you feel horrible because of your immune response responding to that foreign pathogen that's coming in. So you need to get that inflammation, that response to get a really strong immune response, but you have to balance that against safety. And that's why with the current COVID vaccine, you can see they're willing to tolerate injection site reactions, soreness, muscle aches, pain there, a little bit of systemic headache or fever or, you know, achiness for a couple of days, because it's saving people's lives. What they're not willing to tolerate is severe adverse events that, you know, cause people, you know, lifelong conditions or death or other things that are just not acceptable to be given to people that are healthy. Normally healthy. So adjuvants do increase that inflammation at the site. Hopefully the right adjuvant, if you pick the right adjuvant, they'll do it in a very directed way that reduces and minimizes those side effects while maximizing the beneficial effects. There's a question about aluminum salt being toxic. The aluminum, you know, aluminum phosphate, aluminum hydroxide are used in the current vaccines, pretty widely used. They're not thought to be toxic. There have been tons of studies on them. I have my own hypothesis about aluminum salts tend to drive the type of immune response that is similar to an allergic immune response that you usually get against bacteria and some helminths and some pathogens by driving what they call a Th2 type of immune response. I think that the overuse of aluminum salts could lead to you know, the higher incidence of allergies in developed countries where they're used the most. That's a total hypothesis on my part, on things being clean, kids not being exposed to as many bugs, and receiving alum, these Th2 driving adjuvants, might be driving some of the higher incidence of allergy and autoimmune diseases we see in developed countries that don't happen in non-developed countries. But there's not a lot of data to support that. But we're working on allergy also as one of our projects at the university and at Immune, trying to find how these different adjuvants will promote different types of healthy immune responses instead of the Th2 kind of bad response you see when you get an allergy response.
In a related sort of response question, how do folks with autoimmune disorders, I don't know in specific, but like things like lupus or something like that, where you know, how does the vaccine interact with folks with autoimmune issues? And are there cases where that's like a useful thing where you can look for that as a precursor to issues?
No, I don't know if I'd look at it as a precursor to issues. The question is, how well will the vaccine? So most people that have a lot of people that have autoimmune diseases are on medicines that suppress their immune system. So they're more susceptible to COVID. So they're the ones that really need the vaccine because they're more susceptible. But the other question is, how will the vaccine work for someone who's on some steroid treatment or something else that's suppressing their immune system? And I would say that the current clinical data in phase 3 probably doesn't have a high enough N in those numbers of patients with a given steroid or something else they're on to understand how that'll impact their response to the COVID vaccine. And that's where these phase 4 studies will really help define that. Could any of these vaccines cause an autoimmune disease? The answer to that is they could. But so far, I mean, in the millions of doses that have been given of things like the the Cervarix vaccine that includes an adjuvant, the ASO3 adjuvanted vaccine, the Shingrix vaccine, which has an adjuvant that's made down in Hamilton, Montana at the GSK site there. In all of those cases, they haven't seen any increase in the incidence of autoimmune disease in tens of millions of patients. But until you get tens of millions of people vaccinated and you're 5 years out with the COVID vaccine, you won't have a big enough number to say whether you're changing that rate in any meaningful way.
Yeah, that's a tough order, right? I mean,
Yeah,
that's a numbers game in the end.
It is a numbers game.
Yeah. I get there's a question about could you talk a little bit about the AstraZeneca vaccine, which was approved in the UK recently?
I mean, I'm not sure what people want to know about it.
Is it what is it some more? Is it a protein or a protein spike or mRNA? I don't know anything about it.
I would actually have to look. I think that's another adenovirus-based vaccine. I don't think it's a protein. Maybe someone else actually on the call knows. There's like 120 vaccines in different levels of clinical trials.
All right, yeah,
I don't have,
get a pass on that one, I guess.
AstraZeneca one specifically memorized. I don't know if
Right.
it's a recombinant protein or if it's an adenovirus-based vaccine.
But arm soreness is likely a problem.
Probably, and it's not a. It's a good. It means you had a good response against the vaccine.
Absolutely. All right, I'm going to look through here. I think we have one more question from the group, and then we'll turn it over to our sort of the last question of the night. So, what could you speak to? There's sort of a combined. I'm going to combine two questions here, and I hope I get the essence of both. So, once somebody's been vaccinated, and can they still carry the virus, and can they still transmit the virus as if they've been in contact? And does the transmissivity, I guess, go down to zero? And is there still a need for mask wearing and still a need for the social distancing after you have the vaccine?
Yeah, that's. I wish I had a good answer to that. I don't. There are studies ongoing now to see people who have had the vaccine who are protected, can they be carriers? If they then get infected and it's transient, can they be a carrier and give that to somebody else? The answer to that is still out, unknown. Okay. But one thing, if we can get 70-80% of the population vaccinated, then hopefully we don't have to worry about it because you don't see COVID anymore.
Right.
It'll just go away. Maybe you get small seasonal, annual blips in the fall.
Right.
But hopefully we can get it largely controlled. That's the hope. But again, a lot of this is just unknown because remember, a year ago we didn't know anything about this.
Yeah, I mean it's amazing we're asking a lot of the research, that's for sure. So that's a good segue. Your point about getting to herd immunity and up to this 80%. By way of I think general interest and a specific question is is there a I think in a way you gave a really good argument when you bring up the mortality from COVID versus the resistance or the type of people or the level of side effect that may be detrimental to people's health. 30,000 to 3 in terms of the anaphylaxis. That's an impressive and really tangible, concrete comparison. And I mean, that's meaningful to me as a scientist, like, oh man, that's crazy. But in terms of, the general population, there's what I think is a remarkably flummoxing anti-vaccine movement in the country. There's a lot of people who do say, oh, it's way rushed. There's no way I'm putting that, in my body. There's no way they could have done it properly given the rush. And it's, to whatever else is going on, to political schemes that are at play. What is your go-to line to convince somebody to get the vaccine? And then, what are some backup plans? Everybody has the ABCD, right? The backup, contingency. What's your emergency? Like way out there. Give us the talking points that we need to know.
Yeah, it's hard because everybody's different. You have to respect everybody's opinion and feelings on topics like this. There's some people that prior to COVID were part of this anti-vaccine movement. and then no matter what, they don't like, they're against vaccines. They're against a lot of modern medicine, and it doesn't matter what I'll tell that person. You're probably not going to change their mind. You're just going to end up in an argument, and nobody wins. To me, it's the people that they're saying no because they just don't know. They don't have the information. Just like what I showed today. Talk a little bit about you're scared about anaphylaxis. Okay, I get it. That no one has died from anaphylaxis from the COVID vaccine. Knock on wood, hopefully it stays that way. But look how many people would die if they didn't get the COVID vaccine. I mean, it's these things we can manage. Healthcare providers can manage allergic conditions. When I got my COVID vaccine on Friday, they made us all sit there for 15 minutes to make sure no one had a response. The gal at the pharmacy said, I've done, hundreds and hundreds of, over 1,000 of these vaccines now at the pharmacy there on campus, and said, I haven't seen a single case of any case at all. Well, yeah, that's because it's 3 in a million.
They still got their first EpiPen out, right?
Yeah. But they still make you sit there for 15 minutes just in case. And it's as much about public fear as it is about, the reality of actually seeing that in that vaccine center. But the reason they do it is because they don't want to be the healthcare facility that the one person walked out the door, had an anaphylaxis reaction, and died on the sidewalk when they could have been saved by an EpiPen.
Right.
You don't want to be the manufacturer that has that vaccine, because it's going to hit every news station.
Right.
Even if it's 1 in 10 million that happens, you don't want that. So I think education is the key. Teaching people about the safety protocols that are in place to get a vaccine approved, letting them hear from a vaccine researcher or anybody that they didn't cut corners on safety to approve the current vaccine.
Right.
And you're
so I think, the education, right? I think that's the thing is the best response I've heard, like on the spot, from over eavesdropping at the grocery store, listening to a couple folks talk, is one woman was adamantly pro-vaccine and one woman was mostly probably anti, but on the fence. And the woman who was pro didn't judge the other person, the other woman. She just said, I encourage you to read more. And I thought that's a brilliant, easy way to not be a jerk, but to say you're wrong. I encourage you to read more. And that's going to be my go-to line. I encourage you to go to the CDC and not talk about, the CDC of the first year of of the response, but the CDC now, and how the website can really put those things at ease and help you. Yeah, I think that's great. And that's why we were really excited at Science on Tap-Flathead here to have such a powerful voice in the field come on and give us good and solid advice. We have a bit of a tradition at Science on Tap. I don't know if I told you this.
Nope.
Oh boy. I'm not sure you've had the pleasure, but we're going to bring on the director of the biostation to give us some biostation updates and to have the ceremonial last question. So I'm going to hand it over to Jim Elder.
Thank you, Sean, and thank you very much, Jay, for coming to Science on Tap today. I just wanted to say thank you for your heroic work. I remember back in December, when the first results of the Moderna and other vaccine results came in, and I had to sit down for a moment because it really was almost a miracle to hear that data, because people may not realize we could be sitting here right now with nothing in our hands, that there was no guarantee that a vaccine was possible against this coronavirus. And the fact that we have 3, maybe 4, maybe more effective vaccines in our hands right now is an amazing accomplishment. And I want to congratulate you for contributing to that. You're a hero. It's an amazing thing. I want to say that if you ever need a weekend off, your family, come on up. You have a cabin waiting for you at the biostation, and my treat.
Yeah, you haven't fly fished until you fish the Southern Swan River. It's like, sticks everywhere. It's like fishing in the forest. So we'll take you out there.
Yeah, no, that would be great. I have never been, even though I moved over to BMed now on campus, where I was part of DBS for you know, for 4 years, I never got a chance to make it up to the biostation for anything. So I've always wanted to go and see what it was like.
Well, come on up, Jay. And actually, I encourage you, bring your whole team up. I'm not going to cover that completely free. Bring your whole team up with your family. I will guarantee we'll find you a cabin. And
yeah, there might be some travel funds in that, and we'll see.
Anyway, so moving on. So Sean May didn't mention that we have this. I get the pleasure of the last question every Science on Tap. And I was thinking, what should I ask Jay? And usually my questions take the form of, what's your favorite something? And so I was going to ask you, well, what's your favorite virus? Well, that's a weird question. What's your favorite part of the immune system? All right, let's get away from all this virus stuff. We've had too much of it anyway. Then I thought, maybe I'll ask him, Jay, what's your favorite lake in Montana? But that would be a ridiculous question because this is sponsored by the Flathead Lake Biological Station and the Flathead Lakers. So there's only one answer to that question. So I'm not going to ask you that question.
It ain't Fort Peck.
That's right. So I'm going to ask you the following question, which is related to Flathead Lake. I'm sure you've spent some wonderful time at Flathead Lake. What's your favorite thing to do at Flathead Lake? What's your favorite part? Talk about how you've enjoyed the lake during your time in Montana.
So I tell you what, as you saw from the pictures at the beginning, I have been fishing for as long as I could walk. I pretty much fish for everything out there, from deep sea fishing through fly fishing with a fly rod, through doing a lot of bass fishing. And I tell you what, Flathead Lake is a fishery. I know it's controversial because it's non-native, but I tell you what, the smallmouth bass fishery in Flathead Lake is taking off. And it's an amazing fishery. It's an amazing body of water for that type of fishery. As much as people in this part of the state don't like it, it is a warm water fishery. You know, the only way to change that would be to take out the dam, maybe get more flows through there. But there's a good feed base with the perch in there for that kind of fish. So a lot of times when I come up to Flathead, if it's in the main lake, fishing for smallmouth, going up into the Flathead River, into the sloughs, chasing largemouth, spending a weekend up there on the family, I go up there and we'll visit. If we go up to Big Fork, we'll go over visit Echo Lake, do some fishing there. So,
So Sean and Tom Bansack, our associate director, and Sean encourage you to come and take all of the smallmouth bass out of Flathead Lake.
Yeah, like I said, it is a controversial topic because it is a non-native. It's funny, I don't know if you have any data on it, because one of the organizations I belong to is the Montana Bass Federation. And if you go historically back, there's actually records of smallmouth bass being planted at Flathead by FWP. So I don't think it was a legal introduction. I think it was actually FWP planted them in there originally.
I believe it. Largemouth bass, I believe, was the first non-native fish brought to the lake, in fact. So thank you, Jay. Again, just reach out to me. We'll set up a weekend for your family to come up, and I'd love to show you around, and you could take me out fishing and teach me how to catch a smallmouth. Other things at the biostation for folks out there, we're getting back into the swing of things. We are scheduling our summer session right now. We're going to have students on site, COVID willing, and going to go ahead with our summer session. We're having our research interns who are applying right now for our summer research program. Our K-12 program is cranking along. We have a new employee who's helping us with that. And I really looking forward to seeing all of y'all in person as we get the cases down and everyone gets vaccinated and we can meet outside and have a great party once we get past all this. So thank you everyone for supporting the biostation and the Flathead Lakers. And back to you, Sean.
Yeah, I appreciate everybody coming tonight. It was and thanks again, Jay. It was a great talk, and I think that there's just an amazing amount of, I don't know, peace of mind that comes with knowing. I don't know. It's I find being equipped with information is the best way to go about every day, right? And I think that the ability to come on and so clearly. We like science communication here at Science on Tap, if you hadn't noticed, and that was a fine example of it. Stellar work. I'm appreciative and humbled to have you. Thank you very much. I think we currently, again, we're sort of on hold, and hopefully we're going to see how things going. But there's news coming on Science on Tap. That's the best way I can put it. We'll look forward to some news and some interesting changes coming. Hopefully, we'll see. I think that as things develop here, we'll keep you posted on the website. If anybody's dying for something to do tomorrow morning, say around 9 o'clock, you can listen into the KGEZ. All right, yeah, KGEZ, and I'll be talking about giving up an update on our citizen science work around the lake and why I'm not in Antarctica or why I didn't just get back from Antarctica. And I think that you know, it's always fun to be on the radio. Those guys are really good at their job. I mean, hosting Science on Tap's pretty fun, but those guys, they're professionals. I'll leave it there. So I think that's about it, and we really appreciate, again, thank you, Jay, for the wonderful talk, and thank you to the Lakers and Kate and from the biostation for everybody's continued interest, and please stay tuned. Have a great evening.
Get your vaccine and wear a mask.
And listen to Hillary.